Our bodies are made up of millions of cells, each with their own complete set of instructions for making us like a recipe book for the body. This set of instructions is known as our genome and is made up of DNA. Each cell in the body, for example, a skin cell or a liver cell, contains this same set of instructions. The instructions in our genome are made up of DNA. Within DNA is a unique chemical code that guides our growth, development, and health. This code is determined by the order of the four nucleotide bases that make up DNA: adenine, cytosine, guanine, and thymine. So what is genome? A genome is an organism's complete set of DNA, including all of its genes. The term genome was created in 1920 by Hans Winkler. The Oxford Dictionary suggests the name is a blend of the words gene and chromosomes. The human genome is made up of 3.2 billion bases of DNA, but other organisms have different genome sizes. If printed out, the 3.2 billion letters in your genome would take a century to recite. If we recited at one letter per second for 24 hours a day. The Human Genome Project. The Human Genome Project, which was led at the National Institute of Health by the National Human Genome Research Institute, produced a very high quality version of the human genome sequence that is freely available in public databases. The human genome formally began in 1990 and was completed in 2003, 2 years ahead of its original schedule. As researchers learn more about the functions of genes and proteins, this knowledge will have a major impact in the fields of medicine, biotechnology, and the life sciences. The work of the Human Genome Project has allowed researchers to begin to understand the blueprint for building a person. So, what were the goals of the Human Genome Project? To sequence or determine the exact order of nucleotides, the A, T, G, and C, for all of the DNA in a human cell. To determine which sections of DNA represent individual genes or the protein coding units. How it was done. DNA samples are collected from thousands of volunteers. So these samples sent to Human Genome Project centers across the world. Then scientists at centers perform DNA sequencing and analysis. How can we use this information? Better understanding of human disease, personalized medicine and pharmacogenetics, greater insight to cognitive function, insight into human origins, identifying genetic susceptibility to disease. All organisms has a unique genome. For example, a bacteriophage MS2. It is a virus which has a genome size of 3,569 base pairs, and it is the first sequence RNA genome. Bacteriophage MS2 is an icosahedral positive sense single-stranded RNA virus that infects the bacterium E. coli and other members of Enterobacteriaceae. Next is the Haemophilus influenzae. It is a bacterium which has 1,830,000 base pairs, and it is the first genome of a living organism sequenced in 1995. Haemophilus Influenzae is responsible for a wide range of localized and invasive infections. Polychaeus dubium, or the amoeba. It is an amoeboid which has the genome size of 670 billion base pairs. It is the largest known genome, and Polychaeus dubium is a freshwater amoeboid and one of the larger species of protist. Next is the Genliessa tuberosa. It is a plant which has a genome size of 61 million base pairs, and it is the smallest recorded flowering plant genome in 2014. Genliessa tuberosa is a carnivorous species that has a pale bundles or fruit-like organs underground that attract, trap, and digest proteomics. So what is proteomics? Proteomics is the large-scale study of proteomes. A proteome is a set of proteins produced in an organism, system, or biological context. Proteomics enables to understand the structure, function, and interactions of the entire protein content in a specific organism. The proteome is not constant. It differs from cell to cell and changes over time. The terms proteome and proteomics were coined in early 1990s by Mark Wilkins in order to mirror the terms genomics and genome. Proteins can be organized in four structural levels: primary, secondary, tertiary, and quaternary. In primary, the amino acid sequence containing members of a usually 20-unit alphabet. Secondary, local folding of the amino acid sequence into a helicase and B sheets. Tertiary, 3D conformation of the entire amino acid sequence. Quaternary, interaction between multiple small peptides or protein subunits to create a large unit. So why proteins are important? Proteins are responsible for both the structure and the functions of all living things. Genes are simply the instructions for making proteins. It is the proteins that make life. They serve a variety of functions within the cell, and there are thousands of distinct proteins and peptides in almost every organism. Genes make proteins. Cells use the recipes written in your genes to make proteins, just like you use recipes from the cookbook to make dinner. Proteins do much of the work in your cells and your body as a whole. Some proteins give cells their shape and structure. Others help cells carrying oxygen in the blood. DNA creates the different proteins, just as you use different combinations of the same ingredients to make different meals. Proteins are worker molecules that are necessary for virtually every activity in your body. A protein called alpha keratin forms your hair and fingernails and also in the major component of feathers, wool, claws, scales, horns, and hooves. In the blood, the hemoglobin protein carries oxygen in your blood to every part of your body. In your brain and nerves, ion channel proteins control brain signaling by allowing small molecules into and out of the nerve cells. Enzymes in your saliva, stomach, and small intestine are proteins that help you digest food. Cellular construction workers. Huge clusters of proteins form molecular matching that do your cells heavy work, such as copying genes making new protein. In your muscles, muscle proteins called actin and myosin enable all muscular movements from blinking to breathing to rollerblading. Proteins serves as cellular messengers. Receptor proteins stood and outside of your cells and transmit signals to partner proteins on the inside of the cells. The goal of proteomics is to analyze the varying proteomes of an organism at different times in order to highlight differences between them. Proteomics analyzes the structure and function of biological systems. Proteome is quite a bit more complicated than the genome because a single gene can give rise to a number of different proteins through attachment of carbohydrate residues to form glycoproteins. Proteomic methods. Two-dimensional protein electrophoresis, or the 2DE. 2DE is a powerful technique to separate of complex protein mixtures and analyze proteome. Nowadays, the two-dimensional protein electrophoresis is a method of choice for protein separation. This method enables to distinguish up to 10,000 proteins. 2D process. Purify proteins from desired organelle, cell, or tissue. Separate protein mixture in one dimension by isoelectric point. Separate protein mixture in two dimension by molecular weight. Stain gel data analysis. Protein identification by mass spectrometry. So there are three steps in proteomic analysis: the purification of proteins, separation of proteins, and identification of proteins. In purification of proteins, this involves extraction of protein samples from cell tissue or subcellular organelles, followed by purification. In separation of proteins, 2D gel electrophoresis is applied for separation of proteins on the basis of their isoelectric points in one dimension and molecular weight on the other. In identifying the proteins, the separated protein spots on gel are excised and digested in gel by a protease or the trypsin. The eluted peptides are identified using mass spectrometry. The journey from gene to protein is complex and tightly controlled within each cell. It consists of two major steps: transcription and translation. Together, transcription and translation are known as gene expression. During the process of transcription, the information stored in a gene's DNA is transferred to a similar molecule called RNA in the cell nucleus. The type of RNA that contains the information for making a protein is called messenger RNA, or the mRNA, because it carries the information or message from the DNA out of the nucleus into the cytoplasm. Translation. The second step in getting from a gene to a protein takes place in the cytoplasm. The mRNA interacts with a specialized complex called ribosomes, which reads the sequence of mRNA bases. Each sequence of 3 bases called a codon usually codes for one particular amino acid. Transfer RNA assembles the protein, one amino acid at a time. Protein assembly continues until the ribosome encounters a stop codon. So this process is also called the central dogma of molecular biology.